29th, almost Halloween. Another month has passed us by. I'm Michael Kaufman, Dean of the College of Science, and very happy today to have Ivano Aielllo, who is a professor of geological oceanography at Moss Landing Marine Labs and also the current interim director of the Marine Lab. So he's doing a lot of management stuff at the moment. So please have mercy on him. A little background on Ivano before we get started. Ivano got his master's degree in earth sciences from the University of Florence in beautiful Florence, Italy. Then he continued his food tour of education by going to Bologna for his PhD in sedimentology. And I think it must have been rough for Ivano to come from Florence and then Bologna and then to Santa Cruz, because I don't think the food is quite as amazing in Santa Cruz. I mean, there's still really good food, but I think for an Italian, it must be tough to.
You should have seen my luggage. It was just food and wine and I'm serious.
Yeah, I'm sure. Ivano has held a number of positions in engineering, field geology companies, the Italian Geological Survey. He lectured in the Department of Ocean and Marine Sciences at UC Santa Cruz. He was an adjunct faculty member at Moss Landing before getting a tenure track job there in 2006, where he's been ever since. He served as chair of the department, and he does some fascinating interdisciplinary research that looks at the interplay between biological processes and sediments and the formation of ocean bottoms. So we're going to hear all about that stuff today. As usual, if you have a question for Ivano, go ahead and type it in the chat. I'll keep an eye on it. If there's something that it requires me interrupting him, I will do that. Otherwise, I'll let the question hang until the end of the talk where you can ask him yourself. So
right.
Take it away, Ivano.
All right, thanks so much, Michael. Thank you all for coming. And let me say first that, if you want to use a larger version of the screen, meaning there are some graphs that are going to be kind of a high resolution, so you can drop down the window where I am and enlarge the actual displays better for your view. And so the other thing is that my starting slide is, well, it's twofold. One is that you can see the Gulf of California, because we're going to go to the Gulf of California later in the talk. Also because I had to compete with Stephanie Millam's talk from last week. It was fantastic. So I was really intimidated. So there's a space component to that. And one thing I want to add is that when you go out at sea for 2 months in a little tiny can, it's very much like being in space with a lot of ocean around you. And it's a fantastic experience. So I hope I'm going to communicate this feeling with you. So our story today is, as Michael said, it's, well, to me, it's a fascinating story. This is the first time I'm giving this presentation, so bear with me. I hope it comes okay. I just finished preparing a couple minutes ago. And it connects things that apparently can be disconnected, which is primary productivity in the ocean, in the upper left here, to the way an ocean forms and opens. And this is through a series of interesting steps that imply what happens to sediments, marine sediments, once they settle on the ocean seafloor, and not just regular sediments, those special sediments made of tiny, tiny tests of biosiliceous organisms such as diatoms and radiolarians, especially diatoms. And we'll learn a lot about it together. So that's kind of the story for today. It starts with primary productivity, ends up with marine sedimentation, diagenesis, which is the process that turns loose sediments into rocks, essentially. What happens during diagenesis and what happens because of diagenesis underneath the seafloor of the ocean. So we're going to be talking a lot about marine biosilica and diagenesis and biosiliceous microfossils. What are those things? And a good start is a textbook picture of the distribution of marine sediments in the world's ocean. And in particular, if you look at the legend at the bottom here, you'll see that much of the deposition of biosiliceous tests, so those are hard remains, hard parts of organisms such as diatoms, radiolarians, but also sponge spicules, occur not everywhere in the ocean, but in very special places where you have a lot of mixing. So those are the eastern boundary currents and the equatorial divergence currents and all the circular Antarctic belt. So when you have a lot of nutrients coming from the intermediate waters to the surface, and you have light. So that's when plants grow: nutrients, fertilizer, and light. And the zooplankton, which is, the radiolarians, go after the plants to eat them. So where we see a lot of accumulation of biosilica is where you have a lot of productivity at the surface. And there are the reasons, because the ocean seawater is undersaturated in silica. So if you put a little piece of opal in the ocean or a test of a diatom frustule, after a while it will dissolve because of the undersaturation. So the only place where you have a lot of biosilica is where you have a lot of primary productivity. And just to give you a sense, from sediment traps, even in the places that are the most productive, out of 100 tests of diatoms at the surface, only 3 or 4 actually make it all the way to the seafloor. There are two main types of biogenic organisms in the plankton world. As I mentioned earlier, one is radiolaria. They come into shapes, Nacellaria and Spomellaria, and diatoms. They also come into shapes, radial, centric, and elongated pennate. So their test is made of glass. And not only it's a pretty beautiful, intricate, porous test, but also is a mixture of glass and water. And it's very unstable. So after the deposition of these particles and burial, and especially temperature, the glass slowly recrystallizes and turns into a mineral. And the first mineral step in the sequence, diagenetic sequence, is called opal CT, or cristobalite tridymite, which is a pseudomorph of quartz. And that's what it looks like. Those are blades or lattisphere of newly crystallized opal CT. And eventually, with increased temperature, the whole thing turned into microcrystalline quartz. And the original structure, the original valves and tests and remains are completely obliterated. This process is common, obviously, and it characterizes biosiliceous sediments throughout the geological record. Actually, I came here, as Michael said, because I was trying to find better food and wine. And I was coming from this awful place in the Cinque Terre in Italy, where I actually did much of my master work mapping ophiolites. Those are ancient ocean seafloors and crustal units, which have this beautiful sequences of siliceous microcrystalline quartz layers of ancient siliceous deposits made mostly of radiolarians. Here you can see this beautiful ribbon radiolarites from the Jurassic in northern Italy. And if we look at them with a microscope, you see still the remains of radiolarians. This information are key to understand the oceanographic evolution of planet, but also the way oceans form. You don't have to go to Italy, though, to find those things. Actually, right on your backyard, this is a picture I took at Mount Homenhom a couple of weeks ago with my class. This is actually really a few minutes from San Jose. There are radiolarites, and there are radiolarites in San Francisco and Marina Headlands and so on. That's actually why I came here, because I was following the radiolarites. So one way to look at marine sediments is to go up in the mountains. And actually, we like to say that, to look at the deepest marine sediments, you have to go to the tallest peak, or you can go out in the ocean and poke the seafloor and see what's underneath. And that's pretty much what I've been doing with the ocean drilling program, the integrated ocean discovery program in the last about 20 years of my career. This is, probably one of the most fascinating multinational collaboration in terms of exploring our planet. It actually started as a US-only program in the late 50s. The idea was to drill through the Mohorovic discontinuity, and then it evolved into the deep sea program in the late 60s, and eventually changed name several times, but now it's called the IODP, or Integrated Ocean Discovery Program. Drilling was taken out because it's a bad word. But really what it does is drilling around the planet, and all these little dots you see is the constellation of sites that have been drilled in our planet. Pretty much any ocean has been covered, although the oceans are big, 2/3 of the surface. And very much like the space program, actually all the data are available to the public after 1 year of moratorium. So it's an incredible data set of information concerning what's underneath the ocean. And there are very different themes. We look at mostly at the past history of our planet, because the continents are constantly eroding and shedding stuff. The ocean are accumulating. So there are like textbooks of history to read there. So how we do that? We go on cruises with brilliant vessels such as the JOIDES Resolution you see in this picture, which are amazing things that can hang at the surface of the ocean for weeks or months. And using pipes, we just deliver the pipes all the way to the bottom and then drill through. And this ship here, where I spent almost a year of my life, really, it's very big, but so it's about 470 feet, so 150 meters long, but much of it is pipes. There's pipes, pipes, and pipes. So there's a stack where the science is done. There's a science stack. Then there's a deck, and then the place where we, eat and sleep is a really tiny spot. So much of what happens on board is drilling, and the drilling technology is incredibly simple because it's inherited from the 70s. Well, the ship is from the 70s and still works. Just imagine tripping down a pipe from 3 or 4 kilometers from the surface to the bottom of the seafloor by hand, 9-meter segment, and stay in position for weeks so that we can slowly go through the sediments and the rocks. The other interesting part of the story is that the drill bits that we use are very different depending on the type of segments or rocks we're drilling through. So the most used one for what I do is called the Advanced Piston Coring System, which is essentially a piston that's shot into the sediments. This is the shoe of the coring device, while the outside is being drilled. And it collects perfectly preserved sedimentary sequence. You'll see some examples of that. When things get harder, we switch head, we go to extended core barrel, or we actually have to change completely the assembly and go to rotary core barrel. This is for hard rock, like igneous rocks, which we also drill in this story here. The science on board is super exciting. Everything happens as the cores come on board. But this program is old, and we're becoming more and more technologically dependent. This is a beautiful photo I want to share with you. First of all, because the person to the left is my advisor at UC Santa Cruz, Bob Garrison, my mentor and my father in many ways. And the others are some of the founding mothers and fathers of modern marine geology. This picture was taken actually during a cruise to the Mediterranean Sea, when they discovered that the Mediterranean actually dried up in the Miocene. You probably know some of the story here. So it turned into a lake. And just please notice the technology back then: a scale, a Mansell color chart, and coffee. That was it. Now we are much more technologically dependent, although we do a lot of cool stuff on board. We can measure the geochemistry of the pore water, solid phases. We measure physical properties and musical properties. When you're 2 months at sea, you need to find something else to do besides looking at cores. And we are still, though, describing the cores, and that's pretty much what I do when I'm sailing. I use microscopy and just visual description.
Ivano, should we take any piece of information from the fact that the musical picture is the only one with one person in it?
Yeah, it's loud. But good point. Actually, you can play really loud on a ship because it's so noisy that you can hardly tell. Thank you, though. That's a good point. So what happens to the diatoms once they go to bed? Meaning, really, when they get to the seafloor and then they get buried over time. So I'd like to introduce you to 2 of my favorite toys, which are the instruments that we have here in the lab, which are pretty much what supported this research. Those are both San Jose State MRI grants, which were very successful. One is the scanning electron microscope, which uses a very tiny beam of electron to probe the surface of samples. Actually, the cool thing about this photo is that you can see broken up diatoms on the screen when I took the photo. To it is attached an EDX, or energy dispersive X-ray spectroscopy, that essentially uses the characteristic radiation that's produced by the interaction between the accelerated electron beam and the target as a way to measure the elemental composition of the sample itself. So the characteristic radiation we use to interpret the distribution of elements on a sample. The other instrument, it's our brand new X-ray diffractometer, that again uses actually the characteristic radiation produced by a beam of electron bombarding a copper plate. So the K alpha, the radiation coming from that beam, is turned into a monochromatic radiation. And by using a very basic and fundamental principle in physics and mineralogy, which is illustrated by the Bragg's law, the interaction between the wavelength of the incoming radiation, which we know, the angle of incidence, creates a series of diffracted beams, and when the diffracted beams are building a constructed interference, we get a peaking intensity of the diffracted radiation. So there's a relationship between that, the spacing between the planes that make up the atomic structure of a mineral, the lattice, and the angle of incidence. That's called the Bragg law. So that allows us to, first of all, identify minerals using powder diffractometry, and also to probe, to measure the crystallographic spacing within minerals. And you'll see this is really important for our story. Use an example of silica, what it looks like in diffractometric profiles. On the x-axis is the angle of incidence, the 2 theta. On the y-axis is the intensity. And you can see that when silica is glass, like in diatoms, the diffraction is very broad. There's not a distinct peak. And also it's pretty low in intensity. As the crystallization process continues, then the diffraction becomes much narrower, the peaks become more distinct, and the space, because of the spacing between the different planes in the atomic structure. So the other background information that we need to learn together about the story is what we know about how this diagenetic process occurs. So I told you earlier that the glass turns into opal CT and quartz because of temperature. So because of the importance for oil industry of diatomaceous sediments, so just to let you know, much of the oil that's drilled offshore here in California is all from the Monterey Formation of California, which is diatom oozes. So diatoms usually not only bring glass, but also a lot of organic material that then turns into oil. And when they become hard rock, they become both source and reservoirs. So they've been super studied in the 70s and between actually the 60s and the 80s. So what we know as of today is that the range of temperature in which the conversion from opal A amorphous silica to CT occurs is pretty broad. You can find it at pretty low temperature, up to 2 degrees Celsius in the Southern Ocean, up to about 55, 56 degrees Celsius. But much of these measurements are done based on 2 things. Either proxies for temperature, such as delta oxygen 18 stable isotopes in silica, or lab experiments, and very few data are coming from downhole logging, which means finding the conversion at depth and measuring the local temperature. Because you might imagine how hard it is to measure temperatures, in 3,000 meters of water plus 1,500 meters of sediments. But now, as you might guess, we are able to do that. The other thing that we learned about silica diagenesis is that it can be assimilated to a first order kinetic reaction, which goes through a dissolution precipitation process. So the diatom frustose dissolve, the silica is produced as colloids, and which form siloxane bonds, and they actually have slightly negative charges. So they don't coalesce until there's a catalyst, such as a metal hydroxide, for instance, magnesium, which is commonly used to precipitate silica in industrial production, and then the little heart that added up to the mix allows the precipitation of silica. So in natural settings, the previous studies have shown that the opal A, opal CT conversion, which is this line here, on the y-axis is temperature, 45 degrees Celsius to 80. And this is actually the x-axis shows the percent of other stuff that's mixed with, say, diatoms, which is mainly clay. So this means basically that 70% means that 70% of the sediment is actually made of clay. So what is being observed is that if there's clay present in the sediment, this reaction is slightly delayed of a few degrees because, for instance, smectites include magnesium. So the magnesium is actually taken out from the system. So there's less of that catalyst. But the change is only a few degrees. And also notice that how as burial continues and temperature increases, the spacing, the crystallographic spacing in opal CT gets smaller and smaller. Finally, the other important information that we need to know are what other conditions promote or delay opal A to opal CT conversion. And one of them is obviously in any kinetic reaction, the presence of dissolved silica in pore water. So the y-axis has concentration of silica. This is in part per million in pore water. So those are sediments and the pores in the sediment filled with silica. And the x-axis is temperature between 0 and 350 degrees Celsius. So this line here, the blue line, describes the boundary between opal A, amorphous versus crystalline, CT. And as you notice, it takes higher temperature when you have more dissolved silica. Finally, previous reviews of data from the 80s, so those are pretty updated, and I'm hoping to update those reviews, showed that there is a relationship between the timing of transformation from amorphous to crystalline and the thermal history of the sediment. The thermal history is the product of burial rate, so how fast things get buried, and the geothermal gradient. The geothermal gradient is the change in temperature with depth. So, and what is clear is that the smaller the thermal history, the longer it takes for this conversion to occur. So when you have very low temperature and very slow sedimentation, like in the middle of the oceans, like in the gyres, it takes more than 100 million years for this conversion to occur. It still takes millions of years, tens of millions of years, even when you have pretty high thermal history. So only when you get very close to really high, hot and fast sedimenting places, then this whole thing gets closer to, like a million years or so. This is exactly what we're going to be talking about today. What we did, we looked at this really extreme, so to speak, place where you have fast sedimentation, high geothermal gradients, and see what happens there, because we really didn't know. And also, let me add that this is not that unique, because there are places like the Gulf of California, we're going to be going in a minute, the Sea of Japan, the Bering Sea, where these conditions occur. So, but the title of my talk was The Fast and the Beats the Furious. I forgot the title, but what was the fast? So the fast is the Gulf of California and the export of biogenic sediments to the seafloor. It's incredibly fast. This is one of the most productive regions in the world, together with the Bering Sea, the Greenbelt, and the Bering Sea, and the Sea of Japan. So actually, this picture here, which is from 22 years ago, could be taken today, because that's exactly what's happening right now. The North Pacific high just moved to the south. That's why we got rain. So the winds now are blowing to the south. Surface water is pushed out from the Gulf. Deep waters are quelling, and the whole place is blooming like crazy. And when I say blooming like crazy, it means that there are so many dead diatoms getting to the seafloor that every 1,000 years, you get about 1 meter of sediments made of diatoms depositing. Now, it might not sound much, but considering when you go in the open ocean, you get to make 1 meter of sediments, it takes 10 million years. So it's super, super fast sedimentation, accumulation of biosilica, mainly diatoms, and burial, because the whole thing just keeps going down as the ocean opens. Fast. And if you go to Santa Cruz or in Santa Barbara or elsewhere along the coastal California sea cliffs, where I worked for my postdoc at UC Santa Cruz, you see that many of the rock formations there, notice the surfers, are bedded units made of diatoms, which were formed in the Miocene along the California margin when the conditions were very similar to what is now in the Gulf of California. High productivity and a place that was opening up because of the San Andreas Fault system. Which brings me to the Furriers. The Furriers is the Gulf of California. Furriers in many different ways. I had to show at least one geological tectonic map to make it a geology talk. So here's actually the Gulf of California. This is the seafloor bathymetry. You can see the East Pacific rise to the right here, splitting and moving in the Gulf. So the Gulf is a very young oceanic feature, it's about 3-4 million years old. And it's the product of the San Andreas Fault system, which is a right lateral transform fault that links the Eastern Pacific rise at the Rivera Triple Junction to the Pacific rise of Mendocino. And very much like a zipper is opening the continental crust there very rapidly. And why is it furious? So, another textbook figure that actually I learned a new word, furiousist, which you probably have seen in textbooks before, that shows the composition of the upper, of the crust of the mantle. So I mentioned before earlier that temperature increases with that. That's called the geothermal gradient. So if you look at the average geothermal gradients across the planet, in the continents, it takes about 100 kilometers to get to a temperature of about 1,000 degrees Celsius, which means that, it's about 10 degrees per kilometer. So, every kilometer you go deep, it gets hotter 10 degrees. In the oceans, it's higher because the oceanic crust is thinner, and it's about 30 degrees per kilometer, 25, 20, 40, something around that. In the Guaymas Basin, it's more than 500 degrees per kilometer. It's incredibly hot. It gets super hot, very shallow. That's the furiousest thing about that place. And that has tremendous consequence on the way the whole place is working, not just the actual geology of it, but also the biology. That's actually why we went out there with a bunch of microbiologists to see, what happens in the deep biosphere. So going back to our cartoonish understanding of how oceans form, this, and I had a textbook picture of a young ocean opening up. You can see the atmospheric rise happening along the trough here. Those are transform faults. Those are the two continental sides of things, continental margins. And that's actually is a bathymetric picture of the Guaymas Basin. So we are here at the center of the Gulf of California. And you can actually, it's really pretty because you can see the trough here, which is actually split here. You can see the transform fault really clearly, which actually cuts the continental margin like straight. This is actually Isla Tortuga for Baja lovers. And this is Bahia Concepcion. Actually, we go with classes there, with our classes here in Mos Landing. It's kind of fun to be here for 2 months, looking at land and not be able to go and swim. And so our understanding of young oceans was that they form like this. There's a asthenospheric wedge, it rises, push the plate away, and that's where you get magmatism. So in the last few years, there have been a lot of 3D surveys using seismics, which is using sound to look at under the seafloor. And what my colleagues have found is that magmatism is not at all constrained in the graben or the central part of the trough, but actually there are magmatic formations that we call sills. Those are horizontal, semi-horizontal magmatic body extending tens or hundreds of kilometers away from the center. So you can move, here you can see about 50 kilometers away from the axis of the graben. And by the way, the vertical axis is super exaggerated, it's in seconds because you're looking at sound. So you can see these bumps, but they're actually very shallow. I mean, it's all deformed. We, during our cruise, we actually poked in different places that you see here, but the 3 of the places where we worked are the ones that I really like to show you some results of. One of them is right on top of one of the shallowest sills, which is site 1547, actually has beautiful evidence of hydrothermal related colonization of the seafloor with all sort of incredible ecosystem chemosynthetically driven. And then 2 more sites, 46 and 45, one which is right at the edge of the sill and the other one right outside. This is me happy because this was the first core on deck and still using Mansell color charts, which actually reflect really well the coloration of the sediment. And it's so fun because you're out there for 2 months, core on deck. The first core on deck, the whole ship is there. They're all excited. After the third core on deck, you're not going to see anybody anymore. They become boring because not for me though. And the cool thing you can see in this photo, I just zooming in, you can still build burrows. This is actually the seafloor here. And as you go deeper, you get more into the compacted sediment. You can actually see the lamination here. So let me give you a general overview of what we drilled. 3 sites, as I said earlier, the 2, 45 and 46, we went about half a kilometer below the seafloor, 47 only 200 meters. We drilled the sill, so we went across this magmatic body. I mean, this is 100 meters of hard rock. We drill through, we drill above, we drill below, and we recover perfectly preserved sequence of sediments. And we found that indeed in site 1547, the sill is much shallower than it is in site 46. And notice how these 2 sites here are only a few kilometers apart. Here, there's no sill, there's a little tiny extension of it, but the sequence is identical. In other words, so those are again the sites in relation to the seismic profiles. There's an upper part of the sequence, which is about 330 meters on both sides, which is made of diatoms made of glass. Below this depth, the diatoms turn into minerals, into opal CT. And a site 1547, which has a geothermal gradient that's twice as much, so it's much hotter than 46 and 45, this transition occurs much shallower, about twice as shallow, which tells you the temperature is an important factor. Also, please notice how the intrusions, these intrusive bodies, occur within this opal CT zone. In other words, they are overlaying a diagenetic zone that was already present. This is a big part of the story, and I'll give you more information about that. So let's actually now get more into the actual data. And these figures are part of a paper that, finger crossed, one day will be published, I hope soon. They're very complex, so I'll take a few more minutes to go over them with you. So to the left is a plot that shows depth versus 3 different parameters. So the x-axis has 3 different things plotted. One of them is temperature in degrees Celsius, from 0 to 110. The other in darker axis here is silica concentration. This is micromolar, not part per million. And finally, the bottom x-axis is the spacing of the mineral lattice in opal CT. So exactly is how spaced apart are the different layers that make up the opal CT crystals. And it decreases from right to left, which means it becomes more and more crystalline to the left. The symbol represents these different processes. And by the way, this is site 1547, so same symbology, same axis. So the first thing that you notice for sure is that this is the geothermal gradient. So there's actually measurements that have been done in situ. And this shows that the temperature here increases about 220 degrees per kilometer. Here is much shallower, meaning, it increases much faster. So almost more than twice as much. The other thing that you notice is how the opal CT, opal A boundary, the one I showed you earlier, occurs deeper here than occurs here. And finally, you can notice how the sill, this magmatic body, was found inside 1546 at this depth here. And it's about 100 meters thick. Here, it occurs much shallower. So if you look at the data, you see how the pore water silica, which is this red and green line here, red being site 45 and 46, basically identical, increases with depth, indicating diatom dissolution. The diatoms are shedding silica to the pore water system until there's a critical depth in which the whole thing just turns around and the silica start decreasing in the pore water, which means it's actually precipitating in the sediment. And the same thing happens to site 47, just shallower. Right after this, well, a few meters after this change in trend occurs, we start finding the first precipitation of opal CT. The first opal CT precipitating has a larger spacing of the mineral lattice, and as you go deeper, it gets closer and closer, so it becomes more and more crystallized. 2 things to notice here. One is that, first of all, the temperature at which this is happening. If you remember before, our understanding of silica diagenesis was that roughly the highest temperature at which it occurs about 50 degrees, 55 degrees. Here we found it at almost 80 degrees Celsius. That's 30 degrees more than what we knew, which was quite surprising, and it is still very surprising. And 75, 78 degrees in 47. First thing. The second thing is that if you notice, the red dots here are the changes in crystallization and this spacing for site 45 that doesn't have the intrusive sill, but they are pretty much similar to what you see on the adjacent side as an intrusive sill. In other words, the sill does not influence the diagenetic sequence, which is the point I was trying to make earlier, which means that it occurred after diagenesis has occurred. And there's an orange arrow that illustrates that. So shallower sill in 47, where there's a higher geothermal gradient, and the sill intrusion postpones diagenesis. So let's look at the diffractometric profiles. I'm looking at the time. And this is, again, it's a wonderful example of, so this is an X-ray diffractogram showing the profile, the diffractometric profile. This is actually the opal CT forming over time and temperature. So here's a tiny peak that's when the crystals start forming, and then they become more and more prominent until they become really peaky. And the position of the peak actually moves to smaller crystallographic spacing. And again, 77 degrees Celsius where we start finding the first evidence of opacity precipitation. The other cool thing to look at here, because I want to have a little time to talk about the conclusions, is how we can correlate the spacing of the mineral to the in situ temperature. So essentially, we found a proxy for temperature, a paleo-thermometer that works really well, and statistically is very solid, which is also another cool thing. So, but let me show some really cool photographs. So that's what the diatom ooze looks like at 5 degrees Celsius. You know, shallow subseafloor depths. The diatom frastos are perfectly preserved. You can see the smallest detail possible. As you go through the dissolution window and closer to the diagenetic boundary, the diatoms look beat up and they start dissolving. The fine ornaments are almost gone until you get to the place where the pore water silica switches and you start finding neoprecipitates. So what we're calling now opal A prime, which is still amorphous, but it's a autogenic precipitate. So you can see the chewed up diatom. See here a lot of pits, dissolution pits, and the new precipitation here. Finally, the blades start forming. This is the opal CT, and you can kind of guess a few pores here. So there are a few diatoms left in the background, beat up, but those are the newly formed crystalline deposits until you get to the quartz window, and everything is completely obliterated. So now the question you're asking, I'm sure, is how is it possible in such a hot subseafloor, the kinetics of the reaction is so slow? Here comes a little chemistry, which I won't spend too much time talking about it because in the interest of time, since it's 4:40. But let me tell you that you can treat the opal A, opal CT phase change as a first order kinetic reaction. And we are applying existing kinetic parameters using the Arrhenius equation. I'm sure many of you are familiar with this basic chemistry. When you're applying the existing kinetic parameters to what we found empirically, you find that they largely underestimate the temperature formation of opal CT. In other words, we should have expected to find opal CT much shallower than where it is. And this is not just for the Gulf of California, but we actually expanded the study to the Sea of Japan and the Bering Sea. So they're grossly underestimating the temperature formation. So what we did was to recalculate this kinetic parameter based on the empirical data, not lab studies, but actually in situ empirical data. And we came up with a new kinetic parameters, which is namely activation energy and the frequency factor. And these new kinetic parameters match much more closely the actual empirical data, not only in terms of temperature, but also in terms of timing of precipitation. So the bottom line is that the existing kinetic models, which remain existing for the last 50 years, since the 70s, don't reflect kinetics of silica phase change in this type of environments. And the figure that I'm showing you here is similar to the one before, so you can recognize the same parameters. But now here's the model that I worked on, showing predicting the precipitation of opal CT based on the Arrhenius equation kinetics. And when you use the old parameters, you see the intersection between temperature and the actual prediction of temperature at which that will occur will give you an estimate about 50 degrees. Using the new kinetic parameters, we actually are right on target of where we find the actual diagenetic front to occur. So we have a model now that seems to be working, and not just for the Gulf of California. This is another example for site 1547. So in other words, we need to rethink about the kinetics of this reaction. So the question that we need to ask is, why is that? Why is the reaction much slower than what we expected, especially when it's so hot? One other model exercise that I did was to use the kinetics and assume that the sedimentation rates were slower. So the thinking is, what's special about this place is that the burial rate are super fast. So the sediments are really shadowed in the seafloor very rapidly, geologically speaking, but still very rapid, a meter per 1,000 years. So what if I don't change anything else but the sedimentation rate? So what this plot here shows in essence is that when you use the old kinetic parameters, but you actually pretend that the sedimentation was about 20% slower than it is actually in reality. Actually, the old kinetic parameters work. So, which is again telling us that that the burial rates, so to speak, beat the heat. So the fast beats the furious. That's the story. And the other, important story that we already discussed, so I'll go through this pretty quickly, is that the intrusion of this magmatic body seems to be postponing the presence of this diagenetic boundary, which is both fascinating because we thought that much of the diagenesis was actually driven by the intrusion, because this hot magmatic body is about 1000 degrees Celsius. However, what we found, to make a long story short, is that the contact metamorphism is constrained to a very, very narrow zone, only a few meters from the intrusion. When you go away from it, it's like nothing happened. And you can imagine why. Those sediments are full of water. And probably what they do, they kind of contain, so to speak, the intrusion. But the other thing that is really fascinating is that we're seeing the evidence that the diagenetic boundary itself has located the place where the intrusion has occurred. So let me show you a couple of cool photographs. This is what a soft datum looks like. Amorphous silica, actually these varves here represent the seasonal and Paleo-Niño scale variabilities in the Gulf of California. I have 2 students working on this right now, and they'll use that to understand the linear scale variability over the last 500,000 years. Very different story. When those sediments turn into brittle rocks, that's what they look like. They're all broken up, they're hard, and they become really permeable. And if you go to Santa Cruz and near the lighthouse and you look down on the sea cliff, you're going to find very similar things. And you're going to see, for instance, that there, when you have alternation between soft, non-diagenetically mature sediment, hard, brittle sediments, you're going to find a lot of evidence of precipitates coming from fluid traveling through the brittle, more permeable layer. So there are examples of that in the field. So we think that there's a relationship between the opacity, the occurrence of opacity actually controlled the place where magma was rerouted and turned from dikes to sills. So I'm going towards the first part of the conclusion, which is what we learned so far is that in highly productive, fast sedimenting, geothermally hot, it's a mouthful, baby basin, the furious, the rapid burial of siliceous oozes, which is fast, beats the temperature-driven transformation of amorphous silica into crystalline silica. And so we also learned that the actual kinetic models don't explain the kinetics in this type of environment. And also that this transformation creates a major change in physical properties of the sediment, loss of water, the sediment turns from ductile, from soft to brittle, there's compaction, there's a lot of loss of water and pore space. And it essentially affects the routing of fluids, not just magma. That can also be, for instance, the boundary. You can move the boundary between the influence of seawater and the influence of basement fluids, which is very important for biogeochemistry in marine sediments. So I have a model I want to show with you, which takes this idea to a different level. So we have these 2 parameters that we're looking at. One is burial, which is primary productivity. So the more burial means the more productivity, which the more stuff is growing at the surface of the ocean. So here you can see on the left side you have more productive water, more upwelling, more diatoms, and those gray arrows indicate burial rate. So the more productivity, the more burial rate. And then the other parameter is temperature. In this very simple model, I just assume that you have a 1-point source of heat flow, and it's very symmetrical. So somewhere the asthenosphere is shallower here, and there's a gradient that's radial. So basically, on the 2 sides, you find the same temperature. If you go here, but as you move away, the temperature is less and less, okay? So now we go out there with our ship, we're drilling pipe, and we drill 3 places in this ideal diagram. One is 45, 46, which are kind of away from the hot spot, because 47 has more than twice the geothermal gradient. And what we found is that the opal CT is actually shallower in where it's hotter, but also where the sedimentation rate is lower. So this model shows that essentially you can have the same geothermal gradients on both sides. However, where its sedimentation rate is faster, where there's more productivity, the boundary, the opacity boundary, this diagenetic front, is pushed further down. So the faster is the sediment deposited, the more delayed is the reaction. I hope this is clear. So it's not just temperature. In fact, if you start thinking about that, you can play with these two parameters. So you can have places where it's really hot, but there's very low sedimentation rate, or where it's both very, very hot and high sedimentation rate, or the opposite, it's not hot as much and the sedimentation rate is low. That will change the position of this diagenetic front, not just in the Gulf of California, of course, we're talking about globally. And the other idea that we are presenting here is that because of this important physical property change from brittle to soft, magma rising from the asthenosphere actually gets rerouted there. And of course, there's a lot of scientific evidence that supports this besides me telling you that. I mean, we see evidence of that in different types of fluids, and also there are magmatic seals exposed on land that indicate similar processes. The cool thing is that if you think about that, then you can think about why is the Gulf of California doesn't have an oceanic crust? Meaning it does, but on the surface. In other words, anywhere in the Gulf of California, there's no basalts flowing out. They're all stuffed underneath the sediments. And people have been trying to figure out why is that? Why doesn't it open like a cartoon ocean in which you have this volcanic explosions and basalts flowing out? Well, you can imagine that if the ship sank, if the sedimentation rates were much lower than they are, which is actually what happens in most part of the ocean, say 1 order of magnitude lower, probably the opacity boundary would have been much, much shallower. So the magma could have actually been routed all the way near the surface and probably flow out. So that's the connection between primary productivity, 500,000 years ago on the surface of the Gulf of California and the way the ocean is opening right now. And of course, the biogeochemistry of the reaction involved in magma being routed to the surface and the chemicals that come after will have influenced the primary productivity. So that's where the loop is closed. So it's kind of fun to think about that. Also, I had a lot of fun making this animation. And I'm at the end of my conversation with you, but also I'm at my thank you slide, which is, I just took a picture of the poster that's behind me. So those are some of the expeditions. So the first expedition was black and white. So that tells you a lot. And also I had hair. So that tells you the second part of the story. This is actually the team that sailed with me on Expedition 385. It's amazing. It's amazing to be out at sea for so long. It's amazing to work with, so many different people. And actually, that's not everyone on board. That's only the roughnecks, the people, assembling pipes, which is the person with the red shirt, and the scientists and the technicians. There's about 150 people on board. Just a fantastic experience. This is actually one of my certificates because I crossed the equator so many times. And finally, I have the last parting thought, which is what I'm leaving you with. Thank you so much for listening. Oh,
thank you, Ivano. There we go.
That's important.
It is important. Thanks very much, Ivano. All right, I got two questions for you.
Okay.
Question number one is, you alluded to at the end that there's kind of a degeneracy in your models, right? The sedimentation rate and the heat flow, is that right? Yeah. So how can you tell by looking at a core whether the sedimentation rate has changed a lot over time, or whether the heat flow has changed, right? You can't really separate those. Am I correct?
That's correct. Well, so you can reconstruct sedimentation rates for sure. We used to use we're actually doing it right now. Mm-hmm. We use stable isotopes and we correlate the changes in stable isotopes with the global curve. So we get that like to the millennia scale. For the heat flow is a good point. So how did you change your heat flow with time? We don't know that.
Right.
And that's exactly one of the questions. And also because, if I may, this is such a good question because I've been thinking about it a lot. So if you go back to this idea here, you can actually change this too. You can move it to the side. I mean, as the ocean is propagating, so you start cooking a different part. So you're right. You don't know. We don't know. We know what this is now. That's what we can measure. Okay,
cool. And then my other question is kind of a technical question, and that is, how long does it take to put that pipe down that goes down, you know?
It's crazy. These guys are amazing. I had I wanted to show a video how this works.
Mhm.
They can trip down about 10 meters of pipe every, well, it depends on the water depth.
Right.
You're in 3,000 meters of water, 200 makes a big, but they can start drilling a site like this in one day.
That's amazing.
It's crazy. I mean, you've seen this guy, I mean, this gigantic thing. I mean, those pipes are 10 meters each. By hand, they assemble them by hand. I mean, they have,
Right.
and I've seen them doing that in offshore Patagonia. There's a cruise before this. And when the ocean was calm, it was like probably 5 meters swell. That's when it was calm.
Right.
It's just amazing. And the ship stays there. I mean, the hole is this size.
Right.
You
Right.
have kilometers of metal, and the ship stays there because it has, you know, a position system and rotors. Yeah, from the 70s.
Yeah, I was going to say it's not exactly new technology
I
anymore.
miss the 70s. Oh yeah.
Okay, let's see. Do we have any questions from the audience? I don't see any right now. I think they want to get to that glass of wine you were talking about.
That's
Come on,
awesome.
folks. I see some geologists and some biologists and some administrators and some computer scientists on here. Someone must have a
Question.
question for
On
Ivano.
live.
Ah, there we go. So Bill DiVincenzi, Bill, you should be able to turn on your mic. There you go.
Yes.
Hey, nice to see you.
Yeah, Ivano, thank you so much. I got a tour of the facility this morning.
That's
And
right.
I have to say that we have this well-kept secret, this gem that's called the Moss Landing Marine Lab. And I was just very impressed by the beauty of the building, the fact that, as you know, as faculty and residents for sustainability, it's a LEED Gold certified structure, which is very important from a sustainability standpoint. And the research, he keeps one of his employees locked in a cell, which I thought was kind of interesting, but actually, seriously, she's doing work on plastics that are embedded in the ocean, embedded in seafood, and embedded in our bodies, embedded in everything. And she has this very ingenious setup to measure this and conducting, I think, extremely important research. Anyway, I was totally thrilled, and I think that Ivano's done a fantastic job there. So I just wanted to make the comment. I don't have a question.
Yeah, and I'm also sustainable and organic, Bill. I guarantee I'm organic. I'm part of the building. I'm actually part of the building at this point.
Okay, let's see. It looks like Phil Heller has a question. Let's see. Phil, go ahead, Phil.
Can you hear me?
We can.
Ivano, great talk. Really love learning about all this stuff. I'm bummed because I'm at home right now. I couldn't be at the Moss Landing Halloween party because my my COVID booster knocked me flat. So instead, I'll ask my question from here. I was thinking about the photo of the ship with the drill rig on it, and how unstable is the ship because it's carrying that big, tall, heavy rig, and what kind of stabilizers does the ship have?
You know, I thought you were going to ask the question that all the kids ask when they, you know, we give them tour, because, you know, we have a program at sea, because there's a moon pool. So there's a hole in a ship, and they ask, what's this ship not sinking? There's a hole in the middle. That's what you're going to ask. Because it's a good question. There's actually ocean. It's stabilized. Yeah, it's a good question though. So they you know, I'm not very technically savvy, but it fishes down pretty good. There's a system of counterbalance. So there's what is it called? I'm directing a marine lab. I don't even know what those things are called. You know,
Stabilizers?
Well, there's a counterweight. Basically, there's things sticking in the ocean that counterbalances. Yes.
Like
Of course. It's super stable. It's super stable.
I'm glad to hear it.
And also there's a heat compensator. So the whole thing is on springs, and it goes because of the heat. So it goes up and down. Also the tides. You're in the same place for like weeks. The tide changes, and it can change in, you know, meters and meters. So the whole thing adjusts. It's crazy.
Is the pipe actually solidly attached to the boat, or does it slide up and down through the moon pool?
It's maybe for our coffee break, I'm thinking. So it's pulled out from the. So basically, the pipes are, see, they are all stacked in the back of the ship. And then there's a crane that pulls them up and brings them to the rig floor. And then that's where people assemble them one by one. And they're not all the same length because they've been around for that. So everyone, they know each pipe. They have numbers, they write everything by hand. I mean, and really, it's like 70s, you know. So, and the other cool thing, may I tell just a quick story?
Yeah.
There's another ship like this, the Japanese, the Chikyu, the Japanese made 10 years ago, actually got hammered by the Fukushima disaster. But so that's super technological. That thing is a disaster. It breaks every day. They have to helicopter in parts. This thing, you got for 2 months, they can fix anything. Everything can be redone on board because the technology is so simple, but so efficient, you know.
Very nice.
All right.
Okay, we have another one from June Lynn Peoples. June Lynn, you should be able to turn on your mic.
Hey, Julian.
Hey, how are you?
Good. Nice to see you
Thanks for
again.
the presentation. That was fantastic. And really do love Moss Landing. I was just there and recommend that everybody go to visit. I can't wait till it opens up to the public again. But my question really was related to, one, how did you become involved in this research? It's fascinating how one gets to this place. And then the other, what parts of your research are you engaged in related to really the climate impacts and what's happening really more globally in this space?
Yeah, well, thank you for the question. So the answer to the first question is, I got to this randomly, like anything else in life. I was a young PhD student in Florence, Italy, in Bologna, when my professor told me, you need to go to America. I was like, I don't want to go to America. I don't even speak English. And so I did. I came here for a month and I met Bob Garrison, who's, you know, depicted here. And just meeting him changed my life. And also I ended up at Santa Cruz, of all places. So I was just watching movies where everyone was shooting each other and being violent. Then I got to Santa Cruz and it was like the best place I've ever been in. So I stayed. And I finished my PhD here actually. And then I got an opportunity to sail. And, my background was all field work. I gradually met mountains for years before. So the other question is that, so if you look at the record that we collected is an amazing archive of past climate change. So because of the high sedimentation rates, each little tiny lamina you see there is arguably reflecting seasonal scale change back thousands or even hundreds of thousands of years ago. So we can actually understand how the marine productivity, but also the input from land, dust, rain. One of my students is actually looking at wet versus dry periods in the North American continent changed over time. So that's, and we just started. This is going to take years to work, although, as I said, I have 2 students working on it. So, and this story I shared with you, it's more like a bigger picture as how oceans evolve. But of course, it is related to climate change in the sense that, much of the, geologically speaking, much of the carbon dioxide is coming from degassing from underwater volcanoes, indeed. So if they can't degas, you don't get it. So, yeah.
Thank you.
Yeah, thank you. And thank you for attending the talk, Julie. I appreciate that.
Oh, of course.
All right, looks like we got one more from down the hall from you. It looks like Amanda Kahn has a question. Amanda, you should be able to.
It's going to be about sponges. I know.
Turn on your mic.
How did you know my question would be about sponges, Ivano?
Well, guess.
I mean, I'm curious. So in the region where the opal A prime formed, what did the radiolarians or the sponge spicules look like in that same area? Did they also have that same structure as the diatoms?
What's your next question? No, okay, this is a really good question. There's no sponge spicules. I didn't see them yet. But so the point is well taken. So different biosolutions organisms have very different thicknesses in their structure. So spicules are much bigger, and so their diagenetic sequence will be delayed. So what we found is that the dissolution occurs first in diatoms, then in the radiolarians, so to speak, then in the spicules. But there were not really. Couldn't see any radiolarians. Actually, I did see some radiolarians, and they do survive. Actually, you're right. I saw some fragments of the large. I mean, they're still dissolved, but they do survive, more likely. Yeah.
Neat. That would be interesting if like the depth changed at all for where they started transforming into
Yeah.
that.
Yeah, but you know, but the diatoms are so overwhelming there. You know, it's a diatom world in Gulf of California. It's just so you really have to. But we can look into that. That's a really good point. That's a really
It's
good
cool.
point.
Thank you. I learned so much from this talk. That was really great.
Me too, because I just finished this, so I finally learned what the story is about. Thank you.
And therein lies the lesson, right? That you have to give a talk to get your thoughts together.
Yeah, definitely.
All right. Well, we've exhausted the questions, and it's 5 minutes after 5. So I want to thank Ivano again.
Thank you,
Great
guys.
talk, really enjoyable, really informative. And for those of you who are able, you should see a virtual happy hour invitation in your email. All right.
Thanks, everyone. Bye-bye.
All right.
And,
Good stuff.
thank you so much for
Yeah, of course.
hosting. And yeah, I'm gonna do some happy hour. I want to go for a jog.
Yeah, I understand.
So I'm gonna stay because it gets dark so early now. So.
Yeah, I know. No, just do your thing. But if you can come for a few minutes.
No, I will. I mean, for like half an hour, and then I'm gonna switch to shorts and go before the party. We're gonna have a party here tonight.
Yeah, Halloween party?
Yeah.
Good.
Yeah.
Enjoy it.
Yeah. All right. I'll see you on the other one.